It's my pleasure to welcome you to the 2025 Charles L. Cooney Lecture in Chemical Engineering at MIT. The Cooney Lecture was established in 2022 as a premier showcase for rising stars and young talent in the biotechnology arena, bringing emerging leaders in both the academy and industry to campus to discuss aspects of innovation in biotechnology. Before I introduce the individual for whom the lecture is named, as well as our honored lecturer for this afternoon, I want to take a moment to say just a few words about our sponsor, and this is our very own alumnus, Nubar Afeyan. Nubar entered biotechnology during its emergence as an academic field and an industry, completing his doctoral work in biochemical engineering at MIT in 1987. During his career as an inventor, entrepreneur, and CEO, Nubar co-founded and helped build over 50 life science and technology startups. These companies include Moderna, Kodiak, Denali, and many others. He's written numerous scientific publications and is an inventor on more than 80 patents. He's a lecturer at Harvard Business School, and from 2000 to 2016 was also a senior lecturer here at MIT's Sloan School of Management. He teaches and speaks around the world on topics ranging from entrepreneurship, innovation and economic development, to biological engineering, new medicines, and renewable energy. And he is also a member of the MIT Corporation. And now to the individual for whom the lecture is named. Charles L. Cooney is the Robert T. Haslam Professor Emeritus here in our Department of Chemical Engineering. Charlie obtained his bachelor's degree in chemical engineering from the University of Pennsylvania and his master's and PhD degrees in biochemical engineering from MIT. After working briefly at the Squibb Institute for Medical Research, he joined the faculty of MIT as an assistant professor in 1970, and he's been a full professor in the chemical engineering department since 1982. He's received a number of awards, including the 1989 Gold Medal of the Institute of Biotechnologies Award, the Food, Pharmaceutical, and Bioengineering Award from AICHE, that would be Division 15, the James Van Lannen Distinguished Award from the ACS Division of Microbial and Biochemical Technology, also known as the BIOT Division, and he's a founding fellow of the American Institute of Medical and Biochemical Engineers, as well as a fellow of the American Chemical Society. A consultant to multiple biotech and pharmaceutical companies, Charlie sits on the boards of Genzyme, Polypore International, and Biocon Limited in India. His research and teaching span many aspects of biochemical engineering and pharmaceutical manufacturing. He holds more than 30 patents, has published over 250 research papers, and has co-authored or edited 5 books, including Development of Sustainable Bioprocesses, Modeling, and Assessment. In addition to his professional interests, Charlie is a trustee of the Boston Ballet, an overseer of the Boston Symphony Orchestra, and a board member of MIT's Community Service Fund. His other interests include rock climbing, skiing, scuba diving, antique map collecting, and a very picturesque hobby, high-altitude mountaineering. This still scares me every time I see it. But we know Charlie made it back because he's sitting here in the front row, and this is a picture approaching the summit of Ahmedabad in Nepal, 7,000 meters with his son, Matt. Now, this is the part of the program where last year I had a special announcement to make regarding Charlie, where I acknowledged a recent award that he had won. And I think Charlie liked that because now I have another award to announce, which is that in just a matter of over a week, Charlie will be inducted into the National Academy of Engineering, and his citation will read, "For contributions to biochemical and pharmaceutical manufacturing that propelled the establishment and growth of the global biotechnology industry." So I think this is certainly an indication of the high esteem in which Charlie is held, and is a major reason that we have this lecture today. And so now I want to turn to today's lecturer. Markita Landry is an associate professor in the Department of Chemical and Biomolecular Engineering and the Department of Neuroscience at UC Berkeley. She received a BS in chemistry and a BA in physics from the University of North Carolina at Chapel Hill, and her PhD in chemical physics and a certificate in business administration from the University of Illinois at Urbana-Champaign. She also completed an NSF postdoctoral fellowship here in chemical engineering at MIT, working with Michael Strano. Her research centers on the development of synthetic nanoparticle polymer conjugates for imaging neuromodulation in the brain, on the study of lipid nanoparticle cell membrane interactions, and on the delivery of genetic materials into plants. Markita is a member of the scientific advisory boards and a consultant for several major agricultural companies. I just learned she has to go to New Zealand shortly to serve in one of these duties. And she's also a co-founder and CSO of Biophilia Genetics. She has more awards than I could possibly name. I'll list a few of them. She's received more than 40 awards in her career. This includes awards from the Burroughs Wellcome Fund, the Schmidt Foundation, the McKnight Foundation, the DARPA Young Investigator Program, the Beckman Young Investigator Program, an NSF Career Award, and a Sloan Research Fellowship. She's received numerous other prizes recognizing her contributions to science, including the 2022 Vilcek Prize for Creative Promise in Biomedical Science, the 2024 Blavatnik National Prize as a Laureate in Chemistry, and a 2025 Guggenheim Fellowship. And so you'll see there are many photos here that show her awards. I'll note on the far right is a photo from the Blavatnik Award ceremony, and she had so many awards that I didn't even read out the one on the bottom right, which was being recognized as a member of the Talented 12 from Chemical and Engineering News. I'll lastly point out the photo in the top center, which is a picture of Markita with the Strano lab before she left. And so many of you in the room will recognize your academic elders in that photo. And I know Michael's lab is well represented here today. So please join me in welcoming today's Cooney lecturer, Professor Markita Landry.
Well, thank you so much for the invitation, for the introduction. It's absolutely wonderful to be back, especially at a place where I had a significant portion of my training and inspiration for the research program that I run today. And I will start out just by telling you just very broadly and briefly what the entire lab does before digging a little bit deeper into a few specific projects that I've opted to highlight today. So my lab, by and large, is a biotechnology lab. We use the principles of chemistry and physics where I trained to develop tools for translational life sciences applications. And that ranges from understanding transport in plants so that we can deliver biomolecules in the form of genome editing reagents and basically make advances in terms of climate resilient agriculture. What I'll talk about more today, which is neurochemical imaging, so using optically active materials so that we can image the chemistry of the brain and how that relates to some of the behaviors that we are all familiar with in this room. And some side kind of forays and projects into basically taking concepts from colloid science to understand lipid nanoparticle drugs, their interactions with cells, and some recent work in quantifying data distortion in the biotechnology literature. So just as a brief background, I had the privilege of being here at MIT for a seminar a little under 2 years ago, where I talked about a lot of the work that I would say is most translationally relevant in the biotech space. So I'm going to give a very brief update on that work, but I don't want to be redundant. So today I'm going to talk about something very different that I haven't covered in a seminar here at MIT before. So some of the developments that we've made in recent years in the plant biotechnology space have largely borrowed from concepts in human reproduction, assisted reproduction, specifically in vitro fertilization, where you can have really nice control over gamete fusion for the production, essentially, of humans in the lab that then go on to gestate in a recipient parent. Now, we've borrowed from these concepts in the lab to reproduce the benefits of human in vitro fertilization in plant biotechnology, specifically in being able to control fertilization using plant gametes, of course, instead of human gametes, and introduce genome editing reagents, something that is feasible but not ethically possible in humans, but very much tractable and feasible in the agricultural sector. So we've built basically a whole plant in vitro fertilization IVF lab, same exact tools and concepts that have existed for over 45 years in human-assisted reproduction. And this is a snapshot of, again, what we've done essentially since my last seminar here, where we can take an egg cell from, this is a rice plant, and then we can also control fusion with a rice sperm cell to create a rice zygote. So this is a single cell plant. And one of the benefits of having this be a single cell plant is that it takes about 45 minutes before this plant cell starts forming a cell wall, which is the major barrier to exogenous biomolecule delivery. So we can take these rice zygotes and then expose them to genome editing reagents, things like Cas9 or base editors, things that are really, really hard to get into plants, essentially impossible to get into plants without transgenics, right? So doing this in a DNA-free way is really valuable. And we do this in these zygotes, expose them to genome editing nucleases during this critical window when they don't yet have a cell wall, allowing us to increase the efficiency of genome editing efficiencies. And so Any single allele that we target would typically have between a 1% and 5% efficiency using protein-based delivery in a bialistic context, which is kind of the competing technology. In this experiment, what we did is we targeted 4 different parts of the genome simultaneously using 4 different genome editors in a kind of like a one-pot reaction. And because we were able to do this again in this zygote type of system, we just very preliminary data quantified genome editing efficiencies across all alleles with very high efficiencies for every single one. So what this enables or potentiates is simultaneous targeting of multiple alleles within a plant cell in a manner that multiplexing would essentially be unfeasible with individual efficiencies being in the 1% to 5% range, right? So you take a 5% efficiency at 0.05, multiply that or put that to the power of 4 or the number of alleles that you're targeting, and that becomes infinitesimally small. So here we're getting zygotes that are simultaneously edited for multiple targets, this, along with a few other technologies in my lab, has been basically the products that have been used for the development of our company, Biophylogenetics. So basically introducing traits into agricultural crops that require multiple edits in species that have been traditionally recalcitrant to traditional genetic engineering approaches. So that's all I'll say about this work, again, because it's a topic I've covered here before recently. And what I want to focus on today is a bit more on another part of what our lab does, which is neurochemical imaging, why that's important, and some of the contributions that we've made to making brain chemistry visible. So to switch gears here, the brain is a very intricate organ, and it functions on the basis of cell-to-cell communication, as do many other organs in the body, but that's especially critical here in the brain, where very fast interactions between cells gate all higher order function, cognition, and essentially the human experience. Now, I always, as a physicist, learned about neurons as being analogous to something like a circuit where you kind of solder these elements together that allows for electrons to flow, and that's what allows communication transfer in those elements. But unlike an electrical circuit, the brain cells, neurons, don't physically touch each other. So they have to have some other signal transduction that gates information transfer from one active neuron to another neuron or a population of downstream neurons. And that's chemistry. This is essentially chemical engineering. Release of neurochemicals, efflux of neurochemicals, reuptake of neurochemicals in a very efficient way that when it goes wrong, it presents itself in a variety of psychiatric and developmental disorders. And so the importance of brain chemistry is something that I like to illustrate with an example that I hope is familiar to most of you, which is when you're in a Zoom meeting and you have a very clever remark to make and you're trying to get that information across. And so you go on this lengthy diatribe only to realize that you've been muted the whole time. So show of hands, who has this happened to in the past, say, 5 years? Okay, so 98% of the room, then 2% of the room are obviously lying, right? This has happened to every person here. And that's exactly what we're seeing in a lot of our disease indication studies, that the neurons themselves are active. They're able to exhibit what are called action potentials, which essentially physiologically suggests that those neurons are perfectly normal, but they're not actually releasing the neurochemicals that allow you to convey the information to downstream populations. And that's a circuit break. That's just essentially not going to work from a functional perspective. And so that's the part of kind of brain chemistry, right, that we've been really interested in studying. A lot of what we know about the brain is either circuits-based, anatomical, or electrical-based action potentials. We wanted to close the gap by building tools that would allow us to image this chemical element. And I think, again, I argue that that's a perfect problem for a bunch of chemical engineers. And so we went after a few different molecules that serve as signaling molecules called neurotransmitters, or more specifically neuromodulators, because these are molecules that are thought to not only gate information transfer between two neurons, but rather the diffusive component post-release modulates broader networks of neuronal activity. And these are often the chemicals that are more subtle but also precise in the types of behaviors that they help support. And so a lot of these neurochemicals have also not been visible, right? So if you want to solve a transport equation or look at diffusive profiles for these chemicals, you need to be able to see them first, ideally at the levels at which they're being released and then reuptaken by neurons in the brain. So that's, again, precisely what we sought out to do for a few different molecules that have, again, different levels of relevance in terms of disease indications. So the platform that we developed is one that I learned here at MIT based on a semiconductor known as a single-wall carbon nanotube. So this is a molecule, or a particle, I should say, that is highly sensitive to the local dielectric environment and photon recombination. Rather, the ratio between phonons and photons that are produced is highly dependent on the local environment. And so this was an ideal probe for brain chemical imaging. In part because it could be a chemically sensitive probe, but also in part because unlike the probes that I'd worked with as a grad student that tend to photobleach or blink, the stability of the fluorescence was quite nice. And the infrared kind of penetrance of the photons happening between about 1,000 to 1,300 nanometers is also desirable for any sort of biological imaging, especially one that deals with imaging in thick brain tissue. And so borrowing from what I worked on as a postdoc, where I kind of looked at molecular interactions that drove corona phase molecular recognition, here the concept is that you can control the fluorescence of these probes or fluorescence of these materials by pre-functionalizing them with a material that will quench the fluorescence, ideally quench the fluorescence. And then the fluorescence can be recovered in the presence of an analyte that changes the conformation of this polymer in some way, shape, or form, allowing that fluorescence to be recovered. And so it's a very interesting concept because it uses entirely synthetic properties of these materials to generate optically responsive probes, and it's a fundamentally generic platform in the sense that you can, in theory, screen for selectivity, molecular recognition, for any molecular targets of choice. So my very first graduate student, Abraham Bayene, who now runs his own lab and has been doing so for, I think, over 5 years at the Janelia Research Campus, worked on this project and started by having to essentially develop an optical nanosensor for our first neuromodulator target, which was dopamine. And so the way that we do this is initially we screen a large library of polymers, not knowing which are going to have that molecular selectivity for our target molecule. And then we screen them against other molecules that are likely to be found in the brain spaces, right, or the brain regions where we want to take those chemical imaging measurements. So we need selectivity in addition to sensitivity. So we did this for a variety of different polymers, and then we look for, ideally, a turn-on response in the presence of our target molecule, which initially was dopamine, a neuromodulator we wanted to study. And initially, we worked off of some initial kind of hits, and we settled on a repeat of GT, so guanine, thionine, DNA single-stranded nucleotides that formed these rings, so it's a GT6 ring, that ended up working really well for dopamine molecular recognition for reasons that we're still trying to understand. Nonetheless, I'll skip all that work because it's published and it's a little bit kind of early in the lab's career as well. But we took this GT6 carbon nanotube construct that is sensitive and selective for dopamine. And then we collaborated with our colleague at Berkeley, Linda Wilbricht, who is in the psychology department. So she studies development, and she studies also addiction and rodent models of disease. And she was really interested in being able to visualize dopamine in addition to visualizing the neuronal kind of growth and recessions that accompany these diseases. So the experiment looks like this. We take a mouse, we extract brain tissue, and then we can generate what are called these coronal slices. These are a few micron thick slices of brain tissue that we can then use our nanosensor to bathe the extracellular space. So again, neurons in the brain are positioned right within this circuitry to somehow communicate with each other through these chemical signals, and they do so through the extracellular space. So we wanted to essentially paint the extracellular space with our dopamine responsive probes so that we could monitor the diffusive profiles of dopamine release as it occurred within the tissue. And that's exactly what we see in the data. So this is an example data trace, kind of generic, but just to kind of walk you through what to expect. So initially we have our labeled brain tissue. This is a change in fluorescence map. So dark to begin with, we can stimulate either with a bipolar stimulating electrode that recapitulates an action potential, or through an optogenetic stimulation, which is another protein engineering trick you can use to get dopaminergic cells to release dopamine. And you see a bright kind of uptick in terms of dopamine release, and also some variability and heterogeneity, both in the strength and in the spatial distribution of dopamine release. And eventually, through a combination of reuptake through what are called dopamine transporters, proteins that reuptake dopamine for another subsequent signaling event, plus diffusion, we get clearance. And this is a characteristic kind of reuptake constant that we can use to comparatively quantify putative differences in the underlying, for example, dopamine reuptake machinery. Okay, so we can then validate the probe further. This is a video of what that basically static data looks like. We stimulate, you can kind of see release and reuptake of dopamine, which is exciting. If you change the stimulation amplitude, as expected, you get more dopamine released. You can also add what's called the dopamine reuptake blockers. So those basically dopamine transporter proteins can be pharmacologically blocked from reuptaking dopamine. And as you expect, if you add nomifensin, you get more dopamine released and it sticks around for longer, which supports kind of the mechanism of that dopamine reuptake blockage. So all of these kind of measurements just confirm that that what we're seeing is dopamine release. And what was most exciting to us, however, was in the potential for kind of spatial information, right? Bringing some of those concepts about diffusion away from the release site, reuptake kinetics, release site density, and starting to correlate those with actual kind of data in the context of behavior and disease. So the data analysis that I'll be showing for the rest of the talk kind of follows the same analysis guidelines that I will outline here. This is, again, another example video. In this case, it's dopamine release of a labeled brain slice. And what we can do is we can quantify the release site density, right? So within a brain tissue, this is essentially full of neurons, right? But not all of them are releasing dopamine. And we can quantify the proportion of cells of neurons that are actively releasing dopamine. We can also quantify within these release sites how much dopamine is being released per release site, and that has, again, physiologically relevant contexts. And we can also make probes, as I'll briefly talk about today, for other neuromodulator targets to start thinking about which language these neurons are using to communicate. And similar to how we communicate, right now I'm speaking English, right? Neurons speak dopamine, and there are certain neurons that hear dopamine. There are other cell types that speak with oxytocin and other cell types that hear oxytocin. And that's the beautiful way that the brain uses to communicate, right? Brain cells use to communicate with each other. It's through these distinct languages that, as I'll show briefly today, also have a lot of interdependence. And so we've used our probes in a few different contexts. I'll highlight a couple of them here before doing slightly deeper dives into one or two examples. The first study that we did with our dopamine probe involved a more, I'd say, synaptic view of what happens in a neurodegenerative disease called Huntington's disease. This is a monogenic disorder, and it's characterized by mutant Huntington protein expression, which causes plaque formation in the brain, and it's known to compromise dopamine levels overall, because a lot of times the kind of Band-Aid types of drugs that are given to ameliorate symptoms in Huntington's disease are things like L-DOPA or dopamine stimulants, right? So molecules that kind of increase local concentrations of dopamine-like molecules. And so what was really fascinating about this study, and what I really valued about its findings, is that, yes, we saw kind of a decrease in the amount of dopamine in Huntington's disease animals relative to wild type. This was expected. But what was different was not in the fact that there was less dopamine, but rather in why there was less dopamine. There are a lot of different ways that you can have varying levels of neurochemical. It can be in the upstream synthesis of that chemical, and it can be in the ability to release that chemical. It can be in the ability to recycle that chemical for future use. And what we saw is that while the cells in wild types, so genetically normal versus Huntington's disease animals, were very good at releasing dopamine in the first place, what was compromised in the disease situation was in a concept known as release fidelity, right? So you release dopamine once, and that's great. You can track that exact same release site time and time again, And that's what's blunted in disease, right? And the ability to re-release dopamine. So it's not so much a dopamine synthesis or exocytosis problem so much as an issue with being able to essentially recycle dopamine for it to be used in a subsequent signaling event. And again, mechanistically, this is important for things like drug development. We've also taken on, this is actually not our work, this is work by the Lamel and Wilbert labs at Berkeley, but they took on a study looking at the effects of hormones in the dopamine system development. So this is a really interesting study where they removed sex hormones, specifically testosterone in male mice, and then looked at how that affected the development of the dopamine system, which is one of those brain regions that is most plastic or susceptible to formation, especially during adolescence when sex hormones are starting to increase during development. And so what they found was that there was a difference in sham animals versus animals that had been gonadectomized, so had sex hormones removed for puberty, in a brain region called the dorsolateral striatum. So that part of the brain was really dependent on sex hormones to properly develop. But not in another brain region called the nucleus accumbens, where we did not see any differences, basically, in dopamine release capabilities. So just two kind of example studies that kind of used, I'd say, the more molecular features of neurochemical release to draw kind of important biological conclusions. One of the studies that we've most recently undertaken, this is all, by the way, unpublished data, so I'd love to get feedback on this from anyone in the audience who's related to this work or interested in this work. But dopamine has long in the field been considered a encoder of motivation, right? So dopamine is not just a feel-good chemical, but it's almost like it's a reward chemical, where dopamine encodes your ability to study for that exam, even though it sucks right now, but later you'll be happy when you get that A. That's dopamine at work because the reward is not immediate, but it's forthcoming. And that's something that's been known for a while in the dopamine literature, with canonical tests like this one, where you take an animal, you give it a water reward, and you precede that reward with a cue, something like an auditory cue. So you have a bell that rings, and then you get water, and for some reason, mice love water. So a trained animal will hear that cue, and it will get a dopamine spike just when it hears something that predicts a reward, and then a very well-trained animal will essentially get all of its dopamine reward during that auditory signal that predicts the water reward, okay? So basically this is saying, again, you've all been probably well-trained because you're at MIT, that if you study really hard, you're getting your dopamine while you're studying, not when you actually get that A. Does that make sense? Okay. And we were really interested in this kind of reward prediction problem with an interesting subset, which includes that if you are deprived of something, your dopamine reward is even higher, right? So this is actually like a quantitative encoding mechanism, where if you're deprived of water, right, if you're really thirsty, the amount of dopamine that you get with a cue skyrockets, right? And so we were interested to learn more about this in the context of not water or studying, but in the context of the joy that is brought to us by social interactions. This is a study done by Kay Tai's lab, where, again, the details don't matter, but essentially this y-axis measures dopamine, okay? And it measures dopamine in the context of animals that have been allowed to interact with other animals for a long time, versus animals that have essentially been placed in solitary confinement. Okay, so these guys are really deprived of social interactions, and these guys are not. And they are presented with an option to either interact with a novel object. So everyone likes novelty presence. That's usually a small dopamine spike. And then comparing the amount of dopamine reward that animals get depending on whether or not they've been isolated, right? Whether they were deprived of that context. And so as you can see here, both the group animals and the isolated animals have a small dopamine uptick when they are presented with like a little present, novel object, or when they get to spend more time with friends than they had before. But it's really in the isolated animals that get this huge dopamine surge when they're allowed or reintroduced into a social context. And so we were really interested in studying this phenomena, again, from more of like a molecular imaging perspective. And the way we do that is with an animal called a prairie vole. So prairie voles are a really interesting species because they're among the 9% of mammals that have the capacity to form what are called selective peer relationships. They are reproductively monogamous, and they can also form non-reproductive preferences for spending time with other voles, right? Similar to how we have reproductive partners, but we also have the capacity to form non-reproductive, socially constructive relationships, right, with other humans. And so how do we test for sociability? We run something called the partner preference test. So this is a test where you have what's called a partner vole. This is a vole, and this is its friend. So these guys know each other well, okay? This guy is tethered to this part of the box. This is a stranger. These two have never met before. The stranger is tethered to this part of the box, and the focal vole is not tethered. So it can spend time with its friend, it can spend time with a stranger, or it could spend time alone. And we just quantify the amount of time this vole chooses to spend in either of these three compartments. So we were interested in studying this animal because, as I mentioned before, it has the capacity to form non-reproductive preferences for a known partner. And so these are only female voles that are choosing to spend time with another partner versus a stranger. Compare this to mice who don't have a preference. They'd rather spend time alone. And if they spend time huddling with anyone, it's equally split between a partner or a stranger. So it really is a very unique behavior that gives us a window of opportunity to study a lot of both neurotypical behaviors in the context of social interactions and social development, and a lot of also conditions associated with that, including the effects of isolation, and as I'll talk about shortly as well, autism spectrum disorders, where these types of relationships are often compromised in the human condition. So first, we were interested in studying the effects of partner isolation, right? So acute partner isolation on dopamine release. There's no reason to believe that this is a sex-specific effect, and if anything, decades of literature show that reward prediction encodability of dopamine is a universal phenomenon, which of course makes sense. And we were also interested in studying relationship types. So is, for example, there more of a dopamine spike that happens when you're absent of a romantic partner versus a non-romantic partner? And so we studied this using a variant of the partner preference test called the operant test. Here, voles have to actually work to access one of two chambers. So you still have the focal vole here, but it has to like physically press a lever a few times to be able to access its partner, or press a lever a few times to access a novel object. And again, a novel object is kind of like a present, right? There's always this baseline level of happiness that you get with a novel object. And so what we're doing is we're quantifying the amount of effort that these voles put into opening the doors to accessing their partner versus a novel object. And then we're comparing that with a group that's been isolated for 5 days versus a group that's never undergone social isolation. And so this behavior test was done with Annalise Beery, where we just quantified the presses of this lever, and we've quantified this as it relates to total presses, right? So the number of lever presses for the partner over the total amount of presses total. And across both conditions, you can see that before isolation versus after isolation, the voles are much more motivated to access their friend over a novel object, okay? Suggesting that social isolation just causes you to be more motivated to access your friend because you're working harder to open that door. And so we were interested, well, what's going on with dopamine in this context? Pre-isolation versus post-isolation, how is this actually driving, how is dopamine encoding motivation in these contexts? And so what we did was essentially the study that I've already talked about with the analysis that I've already described. And we did this in the context of comparing a few different groups. So we were interested in mate and peer relationships, right? So mate relationships are reproductive in nature. Peer relationships are same-sex relationships, with simplicity for the context of the study. And then also the isolated group, so basically voles that had undergone social isolation versus a control group that had not. Now, Interestingly, we were unable to study male-male peer relationships because if you put male voles in a co-housed environment together, they will actually kill each other. So we were only able to study the female peer interactions and compare those to the mate interactions. So then we quantified the number of release sites, the amount of dopamine released per release site, and the global amount of dopamine released in brain tissues of these voles pre and post isolation. And the first analysis that we did looked at whether or not there was a difference in dopamine signaling across relationship type. Does it matter, your motivation, to access a romantic partner versus a friend? And the answer is no. It seems like across all metrics, the total amount of dopamine released, the number of dopamine release sites, and the amount of dopamine released per release site, these are usually the two most sensitive metrics. In all of these cases, we see that there isn't a difference between mate and peer relationships. So this allows us to group mate and peer data together to better power our study moving forward. So now we were interested in comparing not relationship type, because we didn't see a difference there, but a difference in, for example, sexes. So here, what we're looking at is the amount of dopamine released in total across dozens of animals, looking at the difference between female voles and male voles. So and looking at the differences between isolation and control. So in the female case, we get the predicted increase in dopamine following isolation, right? So female voles that have undergone isolation have more dopamine that motivates them to access a friend, right? More so than a present or a novel object. But we see the opposite, at least trending, right, in male voles. So we dug a little bit deeper into this data by quantifying the density of dopamine release sites and also the amount of dopamine released per release site. And here we started seeing some really interesting trends where, again, female voles showed the predicted increase in dopamine release, as would be predicted, again, by reward encoding following isolation. But we saw very clearly the opposite trend in males. So we're sitting on this data and thinking a little bit about the context in which this means. For context, again, and it's relatively rare to see this stark of a sex dependence in terms of both a behavioral difference and also a neurochemical difference. So we're planning a bunch of more molecular studies to confirm and validate the differences that we're measuring on a neurochemical level. But if they are supported by, again, the mechanistic possibilities for why this might be, including a different number of neurons that are produced, release sites or active release sites, all of these differences being produced by potentially social isolation, right? Then we can maybe better explain how social isolation affects individuals and potentially explain, at least in the context of prairie voles, how this might have a sex dependence. Now, one of the challenges, I think, of working with a synthetic sensor technology is that it's not always possible to make an optical probe for the molecular target that you would like. And so in the early days of my lab, a lot of times we'd be approached by a potential collaborator who doesn't want to image dopamine, but wants to image another molecule. And so it's a little haphazard to screen for molecular recognition when you cannot start with an endogenous molecular recognition element like an antibody or an antibody or something that basically nature has evolved to have recognition for. And so former postdoc in the group, now a faculty member at Pusan National, Sang Hua, worked on this project and developed the concept of evolving molecular recognition for the context of developing new neurochemical probes. And so the context here is that, for example, if we want to develop an optical probe for a new neurochemical like serotonin, instead of testing every single ligand carbon nanotube interaction to see which one gives a positive optical response, we know that a lot of this molecular recognition is DNA-based, and so can we actually start with a large library of DNA oligos, not knowing which are going to be good molecular recognition elements, combine them with carbon nanotubes and our target molecule serotonin, and we know that a molecular interaction is critical for the optical response to happen. And so here, what we can do is we can competitively cause adsorption of sequences to the carbon nanotube that also bind serotonin, and remove sequences that have not bound the carbon nanotube and serotonin together. We can then forcibly recover the sequences that have both bound the carbon nanotube and the serotonin analyte. We can use PCR for amplification of those sequences, and then we can take them into our sequencer to actually identify what those sequences are. And we can do this over the course of multiple cycles until we are able to achieve molecular recognition for the analyte that we started with. And so we can do this, of course, not just with the serotonin context, but also in what's called a control library where we don't have serotonin. This controls for sequences that bind to the carbon nanotube without also having an affinity for serotonin, allowing us to discard those sequences. Also, what this allows us to do is it allows us to start looking at the sequence identities in a non-dimensionalized space, right? So we can, because we've sequenced all of them, take all of the sequences, do some principal component analysis, and when the sequences from the experimental and the control group start diverging, that's when we hypothesize we've achieved molecular recognition for serotonin that we have not necessarily done anything with in the control library. And so that allowed us to basically develop a serotonin sensor. I won't talk about any of our serotonin applied work here just for time reasons, but essentially it's a successful platform in the sense that we can start out with, again, that big round of evolution. We can then take the top 10 sequences from every single evolution round and then experimentally test them for their response to serotonin. And as we would predict, as a function of evolution round from 3, 4, 5, 6, we start getting an increase in serotonin sensitivity where the best performing sensor is the one that's in the last round of this molecular evolution. We do see a slight upward trend in our control group, which would be expected because we are still evolving for better DNA nanotube associations, but far below the sensitivity thresholds of the experimental library. What's really nice about this data too is that you can collaborate. We collaborate with Leila Vukovic's lab at the University of Texas. Give her all our sequences. She's a machine learning expert, and she's told us about sequences that will be really good binders of serotonin, or as I'll show you shortly, oxytocin, without us having to do any experiments. So it's a data set that lends itself well to machine learning-based optimization. We've used this platform to develop what I'll close the talk on, which is molecular recognition of oxytocin. Just to briefly say that we developed a probe recently that allows us to image oxytocin and not its, I would say, largest competitive molecule, vasopressin. So these are called neuropeptides, kind of like dopamine. They signal through the extracellular space. And they differ from each other by only 2 amino acids. So the fact that we're able to image oxytocin over vasopressin was really exciting to us and allowed us to actually use this oxytocin probe to learn some oxytocinergic neurobiology. And that's what I'll close on today, is another study that we just recently published involving the study of oxytocin in basically the formation of social relationships. Oxytocin in the media is known as the love molecule, right? You've probably heard TED Talks or similar contexts where oxytocin is known to be responsible for social bonding, especially in a reproductive context, right? So either between a mate relationship or between a relationship between a parent and a child, usually a mother. And this has been known for quite some time. So one of these earlier studies, again looking at these voles, but in a mate relationship context, included this really interesting assay where they either perfused artificial cerebrospinal fluid, so there's just like a buffer, or they infused oxytocin into these voles. And just this one infusion caused the partners to spend more time with each other. So I remember I saw this data or this study, and I thought, like, I like to think that I have free will in how much time I spend with my husband, but apparently if you just give me an oxytocin infusion, I'll just go home and spend a lot more time with him. And this was, again, just from a physicist's perspective, really fascinating that simple of a manipulation could have such a drastic effect on what I would consider a very complex behavior. This is recent work from the Frumke lab. They did a really interesting study in mice in the context of oxytocin from a reproductive capacity. So mice that have babies, or pups as they're called, will retrieve crying pups, but they'll also retrieve crying pups that are not their own. But virgin mice will hear a crying pup and will completely ignore it. And so this study looked at virgin mice, basically mice that have never had pups, and they used an oxytocin infusion or an optogenetic stimulation of oxytocinergic neurons, and they saw that virgin mice started retrieving pups in the presence of this exogenous oxytocin. So that was a really interesting, again, other manipulation that suggests that something like a maternal gathering behavior could, again, be dictated by the infusion of a specific neurochemical. And I've kind of, I think, experienced this when I used to travel on airplanes before having children. I would put on my headphones, if there was a crying child, and just work on whatever I was working on, and kind of be not bothered, but try to tune it out. Kind of like the virgin mice, I guess, that don't retrieve the crying pups. And then since having children, I just hear a crying baby and I'm flooded with empathy. I don't go retrieve it because that would be socially unacceptable. But I can attest to this phenomenon being true. Whether or not it's due to my oxytocin levels, that remains to be seen. So we were interested in oxytocin's role in a very different kind of relationship called peer relationships that hadn't been well studied before. Peer relationships are, again, non-reproductive in nature, and they're kind of like friendships that you and I form. And it's really hard to study these types of interactions, again, because most mammalian species don't exhibit them. They have, however, a lot of human health relevance. A lack of social connections is one of the primary drivers of premature human mortality. And oxytocin's been implicated or thought to be implicated in autism spectrum disorders. This is a list of, so these are two small-scale clinical trials that looked at intranasal administration of oxytocin that seemed to suggest an amelioration of autism symptoms in children. But then a very large randomized clinical trial recently did the same study and found a null effect. So it's a controversial molecule, at least in terms of oxytocin treatment. So we were interested in looking at oxytocin in the formation of non-reproductive or peer relationships. And again, we come back to our model animal, the prairie vole. And we can run, again, this partner preference test. As a reminder, this is just a physical measurement of how much time this focal animal spends with its friend versus a stranger as a measure for social preferences. Now, what we studied was a really interesting genetic variant of this vole, a transgenic prairie vole from Dev Manoli's lab at UCSF. This vole has had its oxytocin receptors mutated so that they're not functional. So it's basically an oxytocin receptor null mutant or knockout, which means that it's almost like you're taking the ears away from the cells, right? So it's not messing with the capacity to synthesize or release oxytocin, but you're essentially making them unable to understand oxytocin anymore. It's like taking away a language understanding ability, okay? So that's the manipulation that we've done, and we're comparing the two to see how does oxytocin signaling change and how does the behavior change in the context of these non-reproductive bonds. Okay, so what we run is this partner preference test. Again, to recapitulate, how much time does a focal vole spend with a partner versus a stranger? And this was really interesting result that we did not necessarily expect. In the absence of functional oxytocin receptors, which blunt the brain's ability to signal with oxytocin at all, we don't see a very big change in the vole's ability to still prefer a friend. So oxytocin is not dictating the ability for you to form friendships. And this is with what's called a lifetime partner. So these voles have been together their entire lives. It's like your best friend from childhood type of a friend, okay? We reran the assay with what's called a 24-hour partner. Here, this is somebody that you've met yesterday, right? And now you're being given the choice to spend time with them again. The genetically normal animals will choose that friend from yesterday almost always. But now it's the oxytocin deficient animals that are unable to form or recognize or prefer this friend. We can extend the test out to 7 days, and we get the same basically behavior as the lifetime friend. So it's not like oxytocin is dictating the ability to form friendships in the first place, but it's dictating the ability to build a bond that's strong enough for you to quickly be able to recognize somebody as a trustworthy individual. And this is interesting too, because you can measure the number of aggressive bouts. Sometimes these voles go into the stranger chamber and they don't see anyone they recognize and they leave. But sometimes they'll start fighting. But that only happens for the genetically normal animals, not for the receptor null mutant genotype. So what this suggests is that oxytocin is less of a love hormone and less of a social hormone, and maybe a chemical signaling molecule that allows you to define social boundaries between who you trust and know and who you should be careful about, right? The stranger danger type of phenomenon. So I'd be really interested to look at this. We only did this in adult animals, but in the context of development, right? How do people learn, right, to develop social interactions and the benefits that can play? So how is it that friendships can still be formed? We hypothesized that vasopressin might be hijacked as a pathway to compensate for the ability to form friendships. It's a very similar molecule, very similar chemical pathways, also involved very much in like this fight or flight response. So maybe there's a lot of oxytocin produced. KD for oxytocin receptors and oxytocin is high, but not zero for vasopressin receptors. So can we just essentially image oxytocin to debunk or confirm this hypothesis? So again, we used our oxytocin probe in the context I mentioned already. And this is work by Natsumi Komatsu, who's starting her lab at UIUC next year. And then she basically imaged oxytocin release in the brain tissues of these transgenic receptor null mutant voles versus wild-type voles. The high-level takeaway here is that we're not seeing an increase in oxytocin signaling as would be predicted to compensate for the capacity of basically the behavioral assays that we see to still form friendships. If anything, we see less oxytocin release in the receptor null mutant group. And we see that again quantitatively across the metrics of how many release sites there are and how much oxytocin is released per release site. So this was interesting to us because, again, it suggests a suppression of the hormone in the absence of its active signaling pathways. And I think the title of this article nicely sums up the conclusion of the study, which is that oxytocin, again, is not just going to be kind of a love hormone or reproductive hormone or even a friendship hormone, but a hormone that, again, allows us to help define social boundaries and allows us to kind of intuit, right, which relationships are going to be net positive versus net negative for our overall well-being. So moving forward, we're really interested not only in kind of the siloed interactions of a single neurochemical circuit. This is one of my biggest gripes with kind of traditional circuits neuroscience is that you have the dopamine circuit, and that's responsible for motivation. You have serotonin, and that's what goes wrong in depression. And then you have oxytocin and love hormone and bonding. Because neurochemicals release, diffuse, and happen on timescales that are seconds to perhaps minutes long, it's highly likely that these interact in a way that's currently not visible, but that drives a lot of this higher order behavior. So we're interested in making multicolor probes now that would allow us to study more than one neurochemical at a time. And this is, again, just recent preliminary data that we took in our oxytocin receptor null mutant voles. This is the voles that have had their oxytocin receptors messed with. There's no reason that any other neurochemical should be compromised, yet we see a blunting in dopamine signaling here, right, suggesting an interplay between those two neurochemical circuits. So I think it's kind of critical, not just for our lab, for the field, to start considering a multiplexed approach to neurochemical imaging. So lastly, this is the last slide. We're interested in taking this work in many different directions. We're interested in therapeutics. We're working with the molecular therapeutics division to start looking at other candidate kind of molecules that we can use for autism specifically. Interested in a more fundamental way of learning to what extent learning and behavior can be a learned behavior over a innate behavior. And we're working with clinicians at UCSF to get human brain sample tissues to start taking these measurements in human brain tissues and learn about how these neurochemical signaling pathways can go wrong in disease there. So with that, I would, again, greatly like to thank our funding sources, my research group, the slew of people from within the lab and outside the lab that contributed to making these studies possible. My lab and our collaborators come from all over the world, and that's something that we highly value. And I do want to put in a quick plug. I have quite a few recent group members that have recently started labs or will soon be starting labs. So if this line of work is of interest to you, I highly recommend that you reach out to them because they will definitely be hiring a lot of people in the near future. So with that, thank you again. It's an honor to be here. Happy to take questions.
I know I think about prairie voles in a whole different way now. I'm sure others do as well. Questions? Yeah.
Wonderful work. On behalf of all males, I would like to apologize for the behavior of the male mice. I have to, but I wanted to talk, I hope this is not too much of the inside baseball, but your amplification technique, very intriguing. I think there's people in this room that are going to want to try it. But do you think it automatically selects for corona phases that are necessarily facile or weakly bound, right? There's a portion that are not going to be able to be displaced, and so are you missing that parameter space?
That's a good question. I don't think so, because the methods that we use for the separation in that context are quite harsh. So we do a lot of centrifugation. We use buffers that would usually promote desorption. And so in the context of basically being able to select for the sequences that are unbound, if anything, I'm worried about the opposite, about releasing sequences that are good binders, and then we lose them in the unbound category, because we do not sequence those, we do not amplify those. So we're quite selective. The good thing is that when we're starting with 10 to the power of 20 unique sequences, we can afford some loss in the process of the amplification. But we have yet to screen for something that we don't eventually get a hit for. The one thing I will say is that this technique selects for sensitivity, but not necessarily selectivity. So it's good for neuro applications where your selectivity screen is quite generous in terms of being limited, maybe a little bit trickier to engineer the selectivity in there if you're working directly in an intracellular environment or like a blood diagnostic. Yeah.
One of the big challenges in drug development is in depressive illness, bipolar disorder, which are mood and behavioral disorders. I would need a vol model, I suspect, that would be relevant. But how do you see this strategy and tactic playing out in addressing these mood behaviors and depressive behaviors?
Yeah, some of what I've
for
seen,
drug development,
for drug development purposes. Yeah, I think that's a very good point. So there are, for example, a lot of mouse models. I'll use maybe not depression, but autism, because it's a context that I'm more familiar with. But I think the concepts can be translated across conditions. It's a polygenic disease. It's a very environmentally sensitive disease. It's very difficult to diagnose, difficult to treat. And the mouse models that do exist based on human mutations that are known to be strong predisposers of autism, and there's some conversation about that for depression as well, work well in terms of recapitulating some of the, I would say, more mechanics of the disease, so the repetitive behavior, or depressed mice will do what's called a forced swim test, and they will be quicker to sink. But any disease that has a social context to it, right? Depression's a big one. I think social isolation and depression go hand in hand. That becomes trickier to study. And when we do our studies in animals that don't have the capacity to form these social relationships, we don't actually see differences, right? So from a drug screening perspective, I think it's really imperative to choose animal species, models, even cells from the human, right, race, because that's eventually what it's going to go into, and make sure that that the heterogeneity of the response and that the predicted responses kind of fall in line with the mechanism action of that drug. And that's, again, only really possible if it's working in a species that has also co-developed the behaviors that you're looking to treat.
The polygenic disorder, it's a tough problem.
It is. And the genetic approach is more kind of like a one-to-one approach, right, where you're trying to target either the products of a gene, or if you're doing gene therapies, to rectify that gene in the first place. But then the other approach, of course, is to put Band-Aids over the behavior, right? So a lot of what's used now for the treatment of autism, off-label, right, for like depression or other more kind of developmental or psychiatric conditions that treat more the behavior but less the root cause. So that's why I think oxytocin's been such a hot but also debated topic, is because it had thought to be the root cause of autism, like oxytocin deficiency. So we just supplemented it and things will go away. But I think it's not going to be more complicated. It's going to be much more complicated than that. It's more than just the oxytocin circuit that's driving these complex behaviors.
Excellent talk, Markita. I have a question about the data on slide 20, where you're separating the number of release sites into the 3 fidelities. I'm wondering whether you can talk a little bit more about the methodology there and whether that method can be also used for like the oxytocin analysis later or for other molecules.
Yeah, absolutely. For some reason, it's kind of stuck on this slide. Yeah, so there are a lot of nuances that go into this data set, and we have a preprint on it, so I think a lot of the details are there. But basically what we're doing here is testing the sensitivity of dopamine autoreceptors to dopamine release. So here we're actually using a molecule called sulpuride, which is a dopamine receptor antagonist, and what it causes is an increased probability of dopamine release on a per neuron basis. And so we're testing basically after sulpuride addition, how much is that helping dopamine neurons to release dopamine. So in the wild type case, here we basically have, say we're tracking for the sake of argument, just this one release site. We stimulate the brain tissue, this release site releases once. We let it rest for a few minutes, we re-stimulate the tissue, and we look to see if this one release site released again. If it does, that's fidelity 2. We re-stimulate again, if it releases again, that's fidelity 3. Whereas in the Huntington's disease case, we get perfect, essentially similar fidelity for that first release, but once that neuron is asked to speak again, it can't. It's mute, right? The Zoom mute button. So that's kind of the problem that we're seeing in this genotype, is that the ability to release more than one time is what's compromised in the late stage disease condition.
So you need to take a time series of images at a fixed position, and then having the controlled addition of the chemicals at different time points. And
that's right.
Gotcha.
Yeah.
I really enjoyed that talk. I'm wondering if you can maybe comment on how your probes either complement or improve upon other existing chemical neurotransmitter probes, like, for example, genetically encoded probes.
Absolutely. Yeah, I think most of the work I've talked about here is a good example of the advantages, because genetically encoded probes are tractable for use, at least off the shelf, in model organisms. So mice, fruit flies, voles. We're still even working on genetic models for autism, and so it would take years to be able to deploy these probes in non-model species like these meadow voles. And again, I think piggybacking off of the last question or previous question, I think it's really, really important to study the context of, for example, aberrations in social interactions in asocial species. There's so much literature out there on mice that shows no effect, but that doesn't mean that it's not important in our own interactions, and at the end of the day, that's what research is meant to amplify, is the human experience. So that, I would say, is the biggest, I'd say, reason that we've been adamant about developing a technology that's not genetically encoded. That also is a theme that encompasses our genetic engineering work in plants because of regulatory concerns, so very different reason. But we're going into human tissues, and I think that you're never gonna create a transgenic human just for the purposes of looking at chemical imaging. So I'd say that's the biggest motivator for us, in addition to the fact that we're looking essentially at kind of complementary signaling, because we're looking at chemical signaling through the extracellular space, and we're really interested in those kinetics and in those release site densities. We want to be taking those measurements not within cells, but outside of cells. Yeah, great question.
Questions? Oh, there's one there.
Thank you very much. This is really cool work. I was just wondering, during your sensor development, when you're searching through the DNA sequence space, could you just comment on when you go through multiple rounds, what your diversification strategy is?
Yeah, so we don't have an in situ diversification, so it's not like protein engineering or basically random mutagenesis, right, where you're creating diversity in the context of some sort of like pressure, positive or negative pressure. Here, you're starting out with all the diversity you're ever gonna get, but you're starting out with enough diversity that through that competitive kind of complexation process, we know we have to have that perfect kind of trimolecular interaction for molecular recognition to happen. So that allows us to very quickly get rid of many sequences that are either bad binders to the carbon nanotube, bad binders of serotonin, and importantly, both, right? Eventually, we want something that binds all three. So there is no real-time modification other than the diversification of that library getting smaller and smaller to the point where at some round where PCA kind of tells us when to stop evolving, we finally get the sequences that we want to experimentally test in a reasonable throughput manner. So 20 sequences kind of max. In that sense, evolution maybe is a bit of a misnomer, but it's coming more from the diversity of the initial library.
Gotcha. Thank you.
Any other questions? Well, if not, then I have to go over here because there's a photographer. I just want to say again that it's a pleasure to welcome you back to campus. And I think everyone here is able to see something that we always say, which is that chemical engineering provides a foundation to allow you to work in a number of different areas. And so it was very interesting, I know for me, and I'm assuming for many of the students, especially in the audience, to see the different ways that kinetics and transport and thermodynamics and the binding of these polymers to carbon nanotubes can be used to actually probe neuroscience. So thank you very much for your inspiration, and thank you for coming to visit with us.
Of course.